Good morning. Today we'll be revising on the topic ribose nucleic acid. Ribose nucleic acid is mainly found in cytoplasm, little in nucleus. It is present in all organisms, that is both prokaryotes and eukaryotes. It usually occurs as long unbranched polymeric molecule made up of a single chain of polynucleotide. In certain viruses, for example the reoviruses, the RNA is double-stranded and it is the genetic material in some plant viruses and animal viruses. Now the main components of the ribose nucleic acids are the first, that is a sugar. The sugar that is present in the RNA is a ribose sugar with a formula C5H10O5, hence the name ribose nucleic acid. At the carbon 1 position of this ribose sugar, there is a nitrogen base attached. There are 4 nitrogen bases in RNA. They are adenine and guanine and uracil and cytosine. Adenine and guanine, they form the purines, and the uracil and cytosine, they form the pyrimidines. When a nitrogen base gets attached to the sugar at the carbon 1 position, it results in formation of a nucleoside. As there are 4 nitrogen bases, so it results in formation of 4 nucleosides. If adenine is attached, it forms adenosine nucleoside. If guanine is attached, it is guanosine. Similarly, if uracil is attached at the first carbon of ribose sugar, it forms uridine, and when cytosine is attached, it forms cytidine. Now to this nucleoside, if a phosphate molecule gets attached, the phosphate is in form of phosphoric acid, and this gives acidic property to this nucleotide. It results in formation of a nucleotide. So as there are 4 different types of nucleosides, so it results in formation of 4 different types of nucleotides, namely adenosine monophosphate, guanosine monophosphate, uridine monophosphate, and cytidine monophosphate. Now one nucleotide gets attached to another nucleotide through a phosphoester bond. Now already a phosphate is attached at the 5th carbon position by a ester bond, and another bond is established between the 3rd carbon atom of the sugar of the adjacent. Thereby we have this orientation, that is 3' to 5'. So this orientation is actually the phosphodiester bond formed between 2 consecutive nucleotides, and thus it results in formation of a long chain of ribopolynucleotide, which forms the RNA molecule. Now in the previous class we had seen the structure of DNA, so let's compare and find the differences between the DNA and RNA. Now DNA is usually double-stranded polynucleotide chain, whereas RNA is usually single-stranded if it is a non-genetic RNA. Only some genetic RNAs are double-stranded. Now the sugar in DNA is a deoxyribose sugar, whereas the sugar in RNA is a ribose sugar. The nitrogen bases of DNA are adenine, thymine, guanine and cytosine, and here uracil is absent. And the nitrogen bases of RNA are adenine, uracil, guanine and cytosine. Here thymine is absent. DNA, it forms the main hereditary material in all the cells starting from the prokaryotes to eukaryotes. Whereas RNA, it is not a hereditary material in the prokaryotes and eukaryotes, but in some RNA viruses, it acts as the genetic material. Now, the DNA is composed of millions of nucleotides, whereas RNA is comparatively smaller and the number of nucleotides are in thousands. DNA is mainly present in the nucleus and also it is found in mitochondria, plastids and centrioles, whereas RNA is mainly present in the cytoplasm of the cell and a little amount is present in the nucleus. But one should always remember RNA is synthesized on DNA in case of non-genetic RNAs. Now we shall see the types of RNAs. RNAs are broadly classified into two forms, genetic RNA and non-genetic RNA. Genetic RNA is present in plant viruses and animal viruses. They are responsible for carrying the hereditary characters. Hence it is called genetic RNA. Example, the TMV virus, that is the tobacco mosaic virus, is a plant virus, whereas the poliomyelitis, sarcoma, influenza, HIV, coronavirus, these are all animal viruses. The non-genetic RNA are of mainly three types: the messenger RNA, the transfer RNA and ribosomal RNA. Now let us study the structure of the messenger RNA. The messenger RNA is also called as informational or template RNA because it carries the information, the genetic information from the DNA and comes into the cytoplasm where it is translated into a specific protein. The structure of mRNA was first discovered by Volkin in E. coli bacteria, and later it was Jacob and Monod in the year 1961. They coined the term mRNA for it. It forms about 5% of the total cellular RNA, and it is produced by DNA during a process called as transcription and released into the cytoplasm. Now we shall see first the structure of a typical mammalian mRNA. A typical mammalian mRNA has a cap at the 5 end. This cap is composed of a 7-methylguinosine nucleotide. Now this cap is followed by a set of non-coding regions. It is also called as the leader sequence, and then this leader sequence is then followed by a coding sequence. Now this coding sequence has got a start signal and a stop signal. The start signal consists of the initiating codon, either it will be AUG or GUG, and the stop signal is composed of the terminating codon. It will be either UAG, UGA or UAA. Now this coding sequence is then followed by untranscribed regions. It's also called as the trailer sequence. It is a non-coding region, and then this non-coding region is then followed by a poly A tail. Now in prokaryotes, the cap is absent, whereas the leader sequence, the coding sequence, and the trailer sequence and the poly A tail is present in the mRNA of prokaryotes. Now, depending upon the number of coding regions a messenger RNA carries, we have got these two types of mRNAs, monocistronic mRNA and polycistronic mRNA. If a mRNA contains a coding region for a single gene, then we call this as a monocistronic mRNA. Monocistronic mRNA is found in eukaryotes and in prokaryotes also. Polycistronic mRNA, this is a mRNA which carries the coding regions for more than one genes. Now, for example, here you see this is a mRNA which is having a coding region for gene 1, gene 2 and gene 3. So, one mRNA produces 3 types of proteins. So, such mRNA which is having more coding regions for more than one gene, we call this as the polycistronic mRNA, and this polycistronic mRNA is present only in bacteria. Now, sometimes, usually, you see the mRNA is linear, that is, it is form of a straight line. But sometimes in some eukaryotes, the mRNA becomes circular by having an interaction between the 5' end cap and the 3' poly A tail. So this circularization of mRNA promotes recycling of ribosomes on the same messenger RNA, leading to efficient translation. Now, this is the figure showing the circular mRNA where the same ribosome is repeatedly cycled to produce same type of proteins continuously, thereby increasing the efficiency of translation. The second type of RNA is the transfer RNA. This is also called as soluble RNA or adapter RNA. It is called as soluble RNA because it is the smallest RNA molecule, which is composed of about 73 to 93 nucleotides. And it is called as an adapter RNA because it helps as an adapter in forming a connection between the amino acid and the codon region during protein synthesis. Now, transfer RNA also is synthesized in the nucleus by the DNA through transcription. Now, there are almost 61 different types of transfer RNAs based upon their anticodon sites, and these 61 different types of RNAs, they carry the 20 different amino acids which are responsible for the synthesis of proteins. Now we shall see the structure of transfer RNA. Now the structure of transfer RNA was explained by R.W. Holly. This model is known as the cloverleaf model of transfer RNA. According to this model, each transfer RNA has got two ends, the 3' end and the 5' end, At the 5' end, there is this guanosine nucleotide, and at the 3' end, there are 3 free nucleotides, CCA. Now, these CCA end carries the specific amino acid. Hence, this end is known as amino acid binding site. Now, toward this 5' end, here there is a formation of a loop. Now, this loop we call it as the DHU loop, because this loop contains unusual nucleotides, that is dihydrouracil nucleotides, hence the name DHU loop. Now, this loop is responsible for the recognition of the specific enzyme aminoacylate tRNA synthetase. Now, this loop has got a stem here. Now, this stem consists of 4 base pairs. Now, opposite to this DHU loop, there is this T-C loop. Now, this T-C loop has got again an unusual nucleotide, that is the pseudouridine nucleotide, and on the either side of this pseudouridine nucleotide, there is a thymine nucleotide and a cytosine nucleotide. Hence, it is named as T-C loop. This loop also has got a stem. The stem consists of 5 base pairs, and the loop consists of 7 unpaired nucleotides. This loop is responsible for the identification of the ribosomes, the active ribosomes. Now, the loop opposite to the amino acid binding site, we call it as the anticodon loop. Now, this anticodon loop also has got a stem consisting of 5 base pairs, and the loop consists of 7 unpaired nucleotides, and the central or middle 3 nucleotides, they form the anticodons. Now, here you can see this is GGC anticodon, and this anticodon has got complementary pairing with the codons of mRNA, and this transfer RNA with anticodon GGC carries the amino acid alanine. So, like this, the anticodon and the amino acid, they are complementary to each other. Now, this is a secondary structure of transfer RNA. Now, this secondary structure is usually converted into a tertiary structure by hydrogen bond formation between the DHU loop and T-C loop, and forms the tertiary structure of tRNA. So, here you can see this is the DHU loop and this is the T-C loop. They are bound together by hydrogen bonds, and then this is the amino acid binding site, and then this is the anticodon site. So, this forms a L-shaped tertiary structure. Now, the main functions of transfer RNA is: transfer RNA helps in transfer of amino acids from the cytoplasm to the site of protein synthesis, that is ribosomes. It recognizes a specific codon on mRNA to deposit the specific amino acid for the synthesis of polypeptide chain. Now, the third form of RNA is the ribosomal RNA. As the name indicates, it is found in the ribosomes. It constitutes 80% of the total RNA of the cell. It is single-stranded and extremely coiled, folded upon itself in some region within the ribosomes. This RNA is rich in guanine and cytosine bases. Ribosomal RNA and some specific proteins are organized to form the ribosomes. Now here, this slide shows you the two types of ribosomes. That is, in prokaryotes we have the 70S ribosomes, in eukaryotes we have 80S ribosomes. The 70S ribosome has got two subunits. The larger subunit is having a 50S and the smaller subunit is of 30S, where S is the Swedberg's unit, that is the sedimentary coefficient. Now the 50S subunit of the ribosome contains 23S and 5S ribosomal RNA and 31 different types of proteins. The 30S smaller subunit of the prokaryotic ribosome consists of 16S ribosomal RNA and 21 different types of proteins. Now in eukaryotes, the larger subunit is a 60S and the smaller subunit is 40S. The larger subunit is composed of three types of ribosomal RNAs, that is 28S, 5.8S and 5S, and along with the 50 different types of proteins forms the larger subunit of ribosome. The smaller subunit is composed of 18S ribosomal RNA and 33 proteins, and this constitutes the formation of the smaller subunit of ribosome. So thus, the ribosomal RNA forms the main component of ribosomes, and the main function of the ribosomal RNAs, it forms a structural component of ribosomes. It recognizes and binds to the mRNA during protein synthesis, and it maintains the orientation of tRNA amino acid complex during its entry for translation of codons. Now here there is a comparative account of all the three types of RNAs. Now the amount, that is the percentage of the RNAs in a cell, the ribosomal RNA, it forms about the 80% of the total RNAs, mRNA forms about the 5% of RNAs, and the tRNA forms about 15% of the total RNAs. Ribosomal RNA is made up of 120 to 4,500 nucleotides. mRNA is made up of 900 to 1,500 nucleotides, whereas the transfer RNA, which is the smallest form of RNA, it is made up of 73 to 93 nucleotides. Now, ribosomal RNA is highly twisted or coiled, whereas mRNA is linear, and in few cases, in eukaryote, it becomes circular. Whereas transfer RNA, the secondary structure is a clover leaf shaped and the tertiary structure is L shaped. Now if you see the lifespan of these three different has got a short lifespan. Now about the nature of the RNA, the Ribosomal RNA, it is a structural RNA because it forms the structure of the ribosomes. Whereas mRNA and tRNA, they are the functional forms of RNA because mRNA is involved in the function of carrying the messages, and transfer RNA is responsible in transferring the amino acid to the site of protein synthesis. So therefore, these two form the functional RNAs. So the main functions of the three different forms of RNAs: ribosomal RNA is responsible for the structural component of ribosomes. mRNA, it is responsible to carry the genetic information from DNA for protein synthesis, and the main function of transfer RNA is to transfer the amino acid to mRNA codons. So with this, we complete the revision on the ribose nucleic acid. Thank you.